So hello, everyone. I'm Sean, CEO of BioTuring. Welcome to BioTuring Webinar Series, when AI meets biology. So today we have more than 1,000 of scientists registered to attend this talk. And that signals that we have a special guest today. So this is my great pleasure introducing Dr. Rahul Satija. And Dr. Rahul Satija is a scientist who needs no introductions in our community. He's a pioneer scientist in single-cell and spatial biology. And his Seurat algorithms are now being used by thousands of labs in the world. And currently, he's a core faculty member at the New York Genome Center with a joint appointment at the Center for Genomics and System Biology at NYU. And prior to that, Rahul Satija was a researcher at the Broad Institute of Harvard and MIT. And Dr. Satija did his PhD in statistics at Oxford University. And he holds a bachelor degree in biology and music. And I think I have to save the time and leave the stage for Dr. Rahul Satija. Rahul, welcome.
All right, thank you so much for the very kind and generous introduction. And thanks to many of you for joining online and to the BioTuring team for the invitation here. So I'm going to talk about a couple relatively new things from my lab today. The first is I'm going to talk about some computational, a little bit of experimental methods that we developed recently that I hope might be of interest to some of you, whether you're relatively new to the field or whether you've been doing single-cell analysis for a while. And then after that, I want to tell you a relatively recent biological story where we take many of these methods, I think, put them together to discover hopefully something interesting in immunology. All right, so let me go ahead and share my screen. There we go. Okay, and hopefully you can see that, and let's go ahead and get started. So I think one of the really sort of exciting new trends in our field, and presumably many of you are very familiar with this, is the idea of building reference maps or atlases for single cell genomics. And I think this leads to an entirely new and different way for us to be able to analyze single cell data. I think, for many years, many labs, obviously, including ours, we're very focused on doing unsupervised clustering, where we would take data and then sort of pretend we don't know anything about the sample and use unsupervised algorithms to learn clusters and then annotate them based on prior biological knowledge as cell types. That process is long, laborious, takes a lot of sort of manual visualization and annotation. But in principle, can be replaced by reference mapping if reference atlases exist. And it's really sort of an analogy to the field of genomics and really tools like read mappers, like BLAST, that change the way that we analyze genomic data. I joke a lot that I've been doing genomics for 20 years and I've never actually assembled a genome. Every time I've done genomic analysis, I've mapped the reads from my data set onto an established reference. And hopefully either my graduate students or my graduate students will never have to do unsupervised clustering. They'll simply be able to map their data to comprehensive reference atlases. So I think that idea is really quite powerful. Obviously, it's of great interest to the community in general. I think Fabian Theis, who gave a talk at this webinar in the last version, has a tool called SC-Arches for reference mapping, and my lab has developed a number of tools and a web tool called Azimuth that you can go as part of the Human Biomolecular Atlas Consortium that you can go to and just upload your data, and it will automatically map it to one of 13 different references that we have, just like you could do for BLAST for read analysis. So I think all of this is very exciting, and it really changes the way and the speed and the accuracy at which we can analyze single cell data. But the challenge is right now, these references and mapping tools exist almost exclusively for RNA and for single-cell transcriptome data. And our goal very much is to be able to move beyond RNA. We want to understand how chromatin levels, how protein levels, spatial location, lineage, histone modifications, how all of these things affect cellular behavior and identity. But if we want to do that, then it raises a question. How can we build atlases for these other modalities? And this is actually quite a challenge because if we have to build a chromatin atlas and a protein atlas and a methylation atlas and a modification atlas, that will overwhelm the computational community. So what we really would like to be able to do is to build RNA atlases, but then fill in the gaps for all of these other additional modalities as well, to be able to map additional data types onto those maps. And I'm going to show you a strategy that we built for this goal today. All right, so the fundamental challenge, of course, in doing that is that when we want to map one data type onto another, we need to integrate data sets even when they measure different things. And this is the fundamental computational challenge. So here's an example. We've recently built and published as part of Azimuth a hopefully very useful reference of human bone marrows. And we've sort of been very careful, expert curated annotation of multiple different progenitor populations, and also a variety of differentiated progenitors and differentiated lineages as well in human bone marrow. So it's a really fantastic map to trace how gene expression changes during hematopoietic development. But say that we have a tax-seq. Obviously, if you're interested in chromatin, you're going to be interested in, in differentiation, you're going to be interested in chromatin as well. So say that we have a tax-seq data. So for example, this was generated from Will Greenleaf's lab. And we want to annotate these ATAC-seq cells. But ideally, we want to annotate them with the exact same labels and ontology that we use for our RNA reference. We don't want to do a completely independent analysis. But of course, the ATAC-seq data set is built on peaks and not genes. So how do we solve that problem? So my group and others, a few years ago, suggested an initial version of the solution, which was basically to sort of take ATAC-seq data and convert it into fake gene expression levels. So we call these gene activity scores. We can basically take the sum of ATAC-seq reads along a gene, sum them up, and then treat them as a proxy of cellular expression. And then we can integrate those sort of translated activity scores with actual RNA-seq data. And that actually works reasonably well, surprising well, actually. But of course, it makes a very key assumption that in general, open chromatin correlates with active expression. And I think that's a very simplifying assumption. It may hold to some extent in differentiated tissues, but in developing tissues like the bone marrow, where changes in chromatin are so interesting, and also will sometimes precede changes in gene expression, that one-to-one correspondence is obviously broken. So this was really exciting work from Tim Stewart in my lab, but we wanted a new method that would enable us to move beyond those assumptions. All right, so before I tell you how we do that, I want to introduce a sort of an interesting concept called dictionary learning. This is a set of techniques that's very commonly used in image analysis and is becoming increasingly used in genomics. So the idea is that you have a data set, in this case, a noisy image. And what you're going to do is you're going to take a subset of data points in the data set. So, these are little sort of individual patches, and you can see pieces of the tablecloth or pieces of the floor or pieces of the scarf. And we're going to call that subset of patches, we're going to call that a dictionary. Okay. And then what we're going to do is we're going to reconstruct each part of the image as a weighted sum of the dictionary elements. And it turns out that if you do this, you can actually denoise the image. So really to emphasize the essence here, you're learning how to represent a data set as a combination of individual data points. And that collection of individual little elements or data points, that dictionary enables you to denoise the data set. So this is a really sort of cool and powerful approach in statistical learning. The question is, how are we going to apply it in our context? All right, so the idea is that we're going to make use of a third data set. Remember, our goal is to map the single-cell ATAC-seq data set, our query, onto our single-cell RNA-seq reference. And to do that, we're going to use a third data set where we measure ATAC and RNA together. And to do that, we're going to use the 10x multiome technology. Now, the multiome is a little bit more expensive, certainly lower throughput, but it's possible for us to be able to get a few thousand cells. And our goal is to be able to use this sort of multi-omic data set to help facilitate this mapping of ATAC data onto RNA data. And that's where the dictionary learning comes in. So there's a lot of really exciting and I think interesting sort of statistical methods and math in this paper, and I encourage you to read it in detail if you'd like, but I'm just going to really try to emphasize the intuition here. Remember that the sort of essence of dictionary learning is that it wants to represent a data set based on a combination of individual dictionary elements. All right, that's what we're trying to do. So pretend that each cell in the 10x multiome data set is a dictionary element, and that this multiome defines the dictionary, okay? So what we're going to do is we're going to take each single-cell RNA-seq cell, and we're going to represent it as a weighted combination of each of the dictionary elements. And we can do that because both the RNA-seq data set and the multiome data set measure RNA. And then we're going to take the ATAC-seq data set, and we're going to represent each cell there as a weighted combination of dictionary elements. And we can do that because the multiome cells and the ataxic cells both measure chromatin accessibility. So the trick is that after we've done these sort of representations, effectively what we've done is that even though the single-cell RNA-seq reference and the single-cell ataxic query originally measured different things, they can be represented by the same dictionary. They can be translated in the same way. And after that happens, now they're in the same space, and now we can map one of them very seamlessly onto the other. And this really does work very well. So on the left is our azimuth single-cell RNA-seq reference. On the right is our ataxic data set. Same data I showed you before, but what's different now is that we've mapped this data exactly onto the RNA-seq ontology and visualization. So now each single-cell ataxic cell gets a label consistent with one that we had defined in our RNA-seq reference. And so it really is just sort of opening the door for this RNA-seq reference to be able to interpret a new modality as well. I like to use the analogy of an adapter plug. If you go traveling somewhere, you don't want to build an entirely new power system to be able to use your computer. You just want an adapter plug that can allow your existing infrastructure to connect with what's already there. So in a minute, I'm going to show you some benchmarking and some ground truth to convince you of our accuracy, but I do want to highlight kind of one really cool result here. If you look closely, you can see that I've circled a group of cells in the ataxic data set. We've annotated them as ASDC. These are actually, as you can tell, some of the rarest cells in the bone marrow. They're something like 0.002% of all human bone marrow cells. And actually, this is a population that I had helped to discover in my postdoc with Aviv Regev and Chloe Villani and Nirma Cohen. And it's a new type of dendritic cell. It was one of the first discoveries from the Human Cell Atlas, and we found it with single-cell RNA-seq. And again, it's exceptionally rare, something like less than 0.05% of cells in either the blood or the bone marrow. And when we looked at it at the RNA level, it was defined by these markers, AXL and Siglec C6, and so we named them ASDC. We actually don't know, for certain exactly what it is they do, but we do know these markers very well. And what I'm telling you now is that we found these cells in ataxic data for the first time. They're so rare that we would have really struggled to find them in unsupervised analysis. But if we know what we're looking for, we can use this dictionary learning mapping approach. They really pop right out. But how do we know that they really are the same cells? So we can look at this gene, the Siglec C6 marker, and we can find that there is a peak that is uniquely accessible and open only in this axil Siglec C6 population. And in fact, there are hundreds of these enhancers that are uniquely open in this cell population across the genome. And that's starting to give us some hints and some windows into who are the transcriptional regulators of these cells. So it's a cool result, and I'm highlighting it here because this is an example of a population that if we had just done unsupervised attack analysis would have been quite difficult to find, but comes out naturally from an integrated analysis. So most people are using bridge integration to be able to annotate their ataxic cells, but you can do more than that. This is actually a case where we're actually building a continuous pseudotime trajectory, which of course we're very interested in hematopoietic differentiation across the myeloid differentiation lineages. So we can start from hematopoietic stem cells, we go through a couple of intermediate progenitor states, and we end with CD14 monocytes. We can build the trajectory at the RNA level using our single-cell RNA-seq reference. We can build and map the ataxic cells onto the same trajectory, even though they were measured in different cells. And so now we have a matched ataxic trajectory as well, and we can learn a joint pseudotime for these two modalities. And so now we can start trying to explore how changes in RNA relate to changes in chromatin across this differentiation trajectory, even though we measure these modalities separately. And I think that's quite exciting. So here's an example. If you study hematopoiesis, you may know that the gene MPO, or myeloperoxidase, is very associated with commitment to a myeloid lineage. And we can see that gene turns on very sharply and exclusively in myeloid progenitors. But interestingly, the enhancer for this gene, which is a couple kb upstream, that actually turns on in an earlier progenitor stage. And so that's an example of lineage priming, when an earlier progenitor opens a chromatin state prior to expressing the gene. And that sort of lag between RNA and chromatin is actually quite interesting. And also something that would have been difficult, if not impossible, for us to discover if we assumed a one-to-one correspondence between RNA and chromatin. So this is just one example. We look kind of generally across the entire genome and transcriptome. What genes show evidence of this lag? We actually found hundreds of examples, which were highly enriched in cell cycle genes. And so what we found is that the chromatin state of early hematopoietic progenitors has a lot of accessibility for these cell cycle genes, even though those genes are not turned on in early progenitors. And so we think what's happening there is sort of a form of sort of cell cycle priming where these cells are not cycling yet, but they want to be able to turn on and activate that cell cycle expression program at a moment's notice when it's time to differentiate. And that's something that we can learn from this sort of multimodal analysis. All right, so hopefully this is interesting and maybe useful for you if you're generating other modalities, but it does kind of raise the question. You need to be able to have this sort of bridge data set in the middle where we're measuring multiple modalities. So what are the requirements for that data set? Does it have to be massive? Does it have to have thousands of cells? Is a few hundred cells okay? Sort of what are the needs for that bridge data set to be able to facilitate accurate mapping? We did a benchmark analysis with some ground truth data where we sort of slowly downsampled the bridge data set to sort of start to see how that affected algorithmic performance. And what we found is, obviously it depends on sort of the number of the diversity of cell types and the rarity of cell types in your experiment. So for rare cell types, you need to have a little bit of a bigger bridge data set than for abundant cell types, which I think makes a lot of sense. But in general, once your bridge data set has about 50 to 60 cells from each type in your reference, you can get very high levels of mapping accuracy, and you start to saturate pretty quickly after you hit that kind of number. Even if you have 20 cells or less, you still can get high accuracy. And actually, in the example of the ASDC, we only had 4 ASDC in our bridge data set. We were still able to do the mapping accurately. But we start to see failure modes in this regime as well. So, I think if you're doing experimental design, that sort of estimate of having 50 to 60 cells per cell type is a reasonable starting point. And even for technologies like the multiome, a few thousand cells can be quite useful to be able to generate data sets at that scale. Another thing that I want to highlight is I gave an example of RNA and attack, and that's probably the most common use of bridge integration. But there are lots of multi-omic technologies that can be used as bridges. For example, CiteSeq measures RNA and protein together. Single-cell cut and tag Pro does histone modifications in surface proteins. There are technologies for DNA methylation in RNA, even for things like lipid metabolism in RNA. So as these new bridge technologies become available, that enables us to take these RNA atlases that we've used and be able to use them in new contexts, which I think is really quite helpful and exciting. And I'll just give you a couple more examples of that. So this is a data set from Joe Ecker's lab. This is single-cell DNA methylation, which is actually an extremely informative and very high content rich modality. And this is the data set where they did single-cell methylomics of human neurons. And you can see that you get very nice clusters and really high quality data. But it's very difficult to annotate these clusters because we don't necessarily know what these cell types mean because we don't know what genes they're expressing. And so a lot of these clusters don't have good names. We know they're sort of like VIP neurons, but sort of -1 and -2, and we know they're different, but we don't really understand functionally what it is that they're doing. And so what we did is we used that data and the bridge integration technique to map these data onto a single-cell RNA-seq reference from the Allen Brain Atlas. And the value there is there's a really well-defined, clear cell type ontology that's highly interpretable and quite standard in the field. And when we do that, now we can take all of these existing names and convert them into a standardized cell type ontology. So it's not necessarily that we discover a bunch of new cell types, but now we can analyze this data in a consistent framework with other modalities because everything is standardized under a common ontology. We do think that's quite useful. The last example I'll give actually focuses on protein, which is an area of great interest to my lab, and I'll show you lots more examples of that, but to many of you as well, and actually focuses on intracellular proteins. We built a very large atlas of human PBMCs across in COVID data sets from a very large number of studies in patients, and I won't go into the details behind how we do that, but we have a very nice kind of single-cell RNA-seq reference of human blood. But the question is, we know a lot about these cells' inner transcriptomes, but what about their protein levels? And we can use something like CiteSeq to learn about their surface proteins, but can we learn about their intracellular proteins as well? And at least until recently, the availability of being able to generate intracellular protein data alongside RNA has been quite challenging. Now, of course, if we can do something like CyTOF, then we can measure very large numbers of proteins for very large numbers of cells on both the cell surface and the nucleus and the cytoplasm as well. And so what we did is we actually were able to map CyTOF single-cell protein measurements onto our single-cell RNA-seq atlas. And the way that we can do that is we can use site-seq data, where we measure RNA and protein together as a molecular bridge. So we never have to assume that RNA and protein correlate because we're always using that molecular bridge data set in the middle. And this works very, very nicely. So now we can take each of our CyTOF cell profiles, we can annotate it with our high-resolution cell states from single-cell RNA-seq. But that also means that we can take the cell types that we learned from single-cell RNA-seq and we can start to explore their underlying protein expression, both inside and outside the cell, which is quite interesting. So, for example, we can see that inside of toxic T cells, we see a very high expression of intracellular granzyme B, which is a positive control, and is a very nice result as these, of course, this protein is involved in the effector function of these cytotoxic cells. All right, and there's a lot more examples in the paper if you're interested. And you know, I'll just start to wrap up this section and just highlight, you know, a couple of things that are important for bridge integration. The bridge, like I said, does need to be representative of your reference. If you're missing cell types in the bridge, then that can be challenging. If that does happen, then the prediction scores of the algorithm, we showed this in the manuscript, do go very sharply down. So you do get some feedback as a user if something is wrong. But, you know, it's interesting to think about, you know, a question that this approach raises. So, you know, we do need a multi-omic bridge data set to be able to do this analysis. And so from that perspective, like, why not just do multi-omics all the time? Why would you ever measure ATAC on its own if you have the ability to measure RNA and ATAC together? And I think that's a really interesting question. But one of the things that we've realized is that the ability to use this bridge integration approach really gives you a lot of flexibility in your experimental design to really optimize costs, throughput, and data quality. And I'll show you an example of that. So in doing that, I want to mention first a really fantastic technology from Will Greenleaf's lab called NeatSeq. Some of you are familiar with this. It measures intranuclear proteins, RNA, and ATAC all together in the same nuclei. So you get a lot of information. It's trimodal data, but you have to use nuclei, and you have to use the 10x multiome kit. So you pay a price to get these modalities, both in terms of cost, but also very much in terms of data quality and throughput. But it's really exciting to be able to look at these modalities together. Now, this work really enabled my lab immensely, and John Blair, postdoc in my lab, has been developing a technology called PhosphoSeq, and that extends NeatSeq by working on whole cells. So now you can look at intracellular proteins even outside the nucleus, including signaling molecules. But we prefer, you know, to do that, we want to use a single-cell ATAC-seq kit, which gives us much better chromatin accessibility data. It's a lot cheaper and much higher throughput. But then when we do that, we don't get RNA. So the PhosphoSeq technology, as we first published it, just measures intracellular, intranuclear proteins and chromatin accessibility. And the question is, you know, how can we get RNA as well? Just to first convince you that PhosphoSeq works very nicely, you know, we have a really fun collaboration with Barbara Treutlin's lab. They sent us cerebral neuronal organoids derived from HPSC cells. We built a large homemade panel of neuronal and signaling antibodies and ran PhosphoSeq. So we get really, really nice data. It's clearly a very heterogeneous mix of different cell types and different neuronal lineages. In each of those cell types, we get very high quality ATAC profiles from the 10x ATACs kit. We also get very highly informative measurements for proteins, like vimentin, an intracellular protein, phospho-RPS6, which indicates mTOR activity, and OTX2, an intranuclear transcription factor. So all this is great. We don't have RNA. But there are a couple ways that we can get around this. The first is we can use the multi-omic kit and then get RNA as well. But this is actually quite difficult to do and really doesn't return great data. And the reason for that is we're trying to combine so many different technologies and so many different modalities together that getting a single uniform set of protocol steps that works well for all the different permeabilization and fixation requirements really does result in a real drop in quality. And of course, the multi-omic is quite expensive. So what we do instead is we just generated a high-quality single-cell RNA-seq reference from the same set of tissues. We already had our PhosphoSeq data set. And then we did one multi-omic experiment, just on nuclei without any fixation, without any protein, just a regular 10x multiome kind of as a bridge. And what that meant was that each experiment was really run under optimal conditions and returned optimal data quality. And then we can integrate everything together computationally. And that really works beautifully. Now we can annotate each of our ATAC clusters using our RNA-defined ontology. And also we can start building regulatory networks across modalities. We can see how changes in signaling pathways measured by phosphorylated proteins affect chromatin accessibility, and eventually how that regulates downstream gene expression. So I apologize for going through this quickly. This paper is on bioRxiv. But my point is that bridge integration enables us to do multi-omic analysis in a way that I think is quite flexible. We don't have to do every experiment with multi-omics, but just a subset. And that enables us to really retain the best quality data, and I think at the optimal price and with a very flexible experimental design. All right. So now I want to switch gears a little bit and talk about a biology story that was a collaboration between my lab and Dan Littman's lab, and also leverages samples from the NYU Vaccine Center. This work has been led by Bingji and Robbie, who are our postdocs in my lab and Dan's. And the goal of this was to better understand T cell responses to COVID vaccination. So as of course many of you know, understanding T cell responses to vaccination and infection is a really key and exciting area in immunology. I think most of the time when we think about the efficacy of vaccines, we think about antibodies and antibody titers, and those are of course essential and important as well. But there's been, really a significant recognition in the community that over time that T cell contributions are also required for early and in particular for durable protection against SARS-CoV-2. And especially against things like new variants and things like long-term immunity, even after the antibody titers have decreased. And so there's a recognition in the community, though, that while it's very easy to measure antibody titers, it's a lot more difficult to be able to identify, characterize, and track T cell infection, and we need new molecular assays to be able to do that. John Wherry, who's a leader in this field, actually wrote an editorial talking about, different types of assays that could be used to understand and characterize T cell responses in both vaccination and infection. One of the things I found fascinating about this article is it makes no mention of single cell sequencing, despite that you would think that single cell sequencing would be a very powerful approach to be able to study the responses of individual T cells. And then one of the reasons that the article doesn't talk about single cell sequencing very much is that, as many of you know, single cell sequencing traditionally has not been a particularly effective or high resolution tool for being able to study T cells. T cells are small, they have limited RNA content, and what we know, they have substantial functional diversity by looking at their protein levels. Often when you look at them in single cell RNA-seq data, they just form these sort of blobs, and we're unable to resolve sort of the true level of diversity that we know is functionally present in a collection of T or lymphoid cells. And so actually there have been a number of papers that studied COVID vaccinated and infected patients, and in particular vaccinated patients. But really all of those studies failed to identify specific subsets of T cells that were being induced by vaccination, even though we know that, of course, that they're there. So that raised a really interesting question of, where are these cells? Can we find them, and can we figure out what makes them unique and functionally valuable? And that's what we set out to do. All right, so our data set consisted of 6 individuals, and this was like the first round of Pfizer vaccination, if you remember this, for a few years ago. So you would have your first shot. We have day 0 before the first shot, and then you would have your first shot, and then 3 weeks later you would have your boost vaccination. So we have samples at day 0, day 2, day 10, and then 1 week after boost at day 28. We ran a couple of different technologies here on these samples to generate, I think, a reasonably large data set. The first is we ran 3' single cell RNA-seq, but we actually ran it with cell surface proteins using the CyC technology developed here at the Neurogenome Center. And we also ran a chromatin accessibility profile in a separate experiment, although on the same samples. But we ran ASAP-seq, which enables us to measure chromatin accessibility and protein at the same time in the same samples. And we always use this sort of panel of 175 cell surface proteins for the site and ASAP-seq experiments. We did some additional modalities, and I'll show you those in a minute, but this was kind of our initial starting data set. And our goal was to find, ideally, T cells that were responsive and antigen-specific to this course of vaccination. All right, so this is a UMAP visualization of our data. You can see that this is very high-quality site-seq data. It's all publicly available. And we cluster the data using the weighted nearest neighbor algorithm developed in my lab as part of Seurat v4, which is well suited for characterizing data where we get multiple modalities from single cells. So as a result of this data and analysis, we get really high resolution, I think, onto different T cell subsets. These T cells are clearly not blobs. They're showing extensive levels of diversity. And so there are a lot of different cell types in this analysis. But really, I'm going to spend most of the talk focusing on this little group right here. And the reason that I'm going to focus on this little group is we think these are the cells that we're looking for, and they're quite interested in, and that comes out even just by splitting this analysis across the different time points that we have. You can see there's a very small number of these cells at day 0. Those may be patients that had prior exposure to COVID prior to vaccination. But the abundance of these cells really jumps after the first shot, but particularly after the boost vaccine, when we start to see them extensively at day 28. So this is the population that we started to focus on. Now, remember that we do have deep RNA-seq and protein data available for each of these cells, so we can discover biomarkers across multiple molecular modalities, which is exciting. What we can see at the protein level is that there's very strong expression of biomarkers that have been canonically associated with T cell memory precursors, an activated T cell, including CD38, ICOS, and HLA-DR. But we can also see, for the first time, a very clean RNA signature of a subset of about 200 genes that is uniquely and specifically expressed in these clusters of cells. And in fact, there's actually two different clusters, one of which is expressing cell cycle genes and appears to be highly proliferative, and the other one, which is not. That's valuable for two reasons, actually. I think people have traditionally assumed that these cells are uniformly proliferative, but we actually see two different populations at roughly equal frequency. And also the fact that we have both proliferative and non-proliferative populations enables us to separate their gene expression signature into two different parts, one of which is shared between these two populations and one of which is unique. And actually this shared signature we think is quite valuable for being able to identify these cells in new data sets, as I will show you repeatedly later in the talk. All right, so that's how we can find these cells in single-cell RNA-seq. We found them because we had very high-quality protein data as well, and that helped us separate out this population. But what about an attack-seq analysis? Because I said that we also generated chromatin accessibility data paired with cell surface proteins. So we can do an unsupervised analysis. We can analyze and cluster the single-cell attack-seq data, as we do here on the left. And again, we get these blobs of T cells, and it's very difficult for us to be able to identify which of them, if any, is related to the specific vaccine-induced group that we discovered previously. But we can map this data to our site-seq reference using bridge integration, the same computational method that I showed you before. That previous data measures RNA, this measures attack, but we can use actually a publicly available data set from 10x Multiome, just on their website, where RNA and attack are measured together as a molecular bridge. And now, when we analyze the attack-seq data set with bridge integration, we see a subpopulation here, which maps precisely to the vaccine-induced group that I showed you in the previous slide. Okay, so this is a computational prediction, right? We identify a population of interest in site-seq data, and now I'm claiming, using some bridge algorithm, that we found the same population in attack-seq. Now, they are in the same place in the UMAP, but how do we know they're actually the same cells? How can I actually convince you that this is a valid mapping that we've made here? This is where the second modality becomes so useful. So when we did the bridge integration procedure, we withheld any information about cell surface proteins from the analysis, but we measured them in the same experiment. So then we looked at the cells that we identified in attack data as mapping to our vaccine-induced population, and we said, what protein biomarkers do they have? If they're the same cells, they should have the same protein biomarkers as we had previously discovered in the RNA data. And that's, in fact, exactly what we see. We see elevated expression of CD38, CD278, and HLA-DR specifically and uniquely in this cell population, as we had already validated to be true in the site-seq data. So that's a really important sort of validation for us to be confident in this mapping. The other thing that we see, which is really quite nice, is now that we have chromatin accessibility profiles for these cells, just like I showed you with ASDC before, we can start to look for enhancers that are uniquely and specifically accessible in only this rare cell population. You can find some quite interesting candidates. So, for example, CD38 and ICOS are two markers of this population. And again, we can find enhancers that are uniquely and specifically accessible only in these cells. And actually, we can find about a couple thousand of these enhancers genome-wide. It turns out that actually 16 of those enhancers house SNPs, or single nucleotide polymorphisms, that govern or that are associated with susceptibility to the ability to fight off COVID. So we don't know if we haven't done any downstream functional validation, but it's an intriguing hypothesis that maybe their location and their expression, their accessibility, particularly in these cells, is contributing to that phenotype. The other thing that we can do if we have lots of these enhancers is we can look for transcriptional regulators that are now responsible for establishing and maintaining this cell state by using motif analysis, which is quite common for attack data. And so when we do that, we get a very nice and strong hit for the transcription factor BATF3. So that leads to a prediction that in humans, BATF3 is responsible for being able to generate and drive the maintenance of this cell state. Turns out that actually right around the same time that this paper, we made this discovery, a paper was published in mouse showing that BATF3 programs CD8 T cell memory. And so I think our work is very consistent, showing that's likely true in humans as well. All right, so at this point, I've shown you a lot of data from multiple different molecular modalities on what I'm calling vaccine-induced cells. But I have not actually said that they're antigen specific. I've just said that they increase in frequency during the vaccine response. And there are two different reasons for why they could be increasing in frequency. The first reason is that maybe they really are antigen specific. Maybe they are responding to the vaccine and proliferating as they should. That's a good possibility. But the other possibility is that they're passenger responses. So for example, after vaccination, the microenvironment, there's a lot of inflammation, a lot of cytokine signaling. So maybe these cells are just proliferating in a non-antigen specific way as a result of the broader changes in the microenvironment. In our initial data set, we actually can't be sure whether these are antigen-specific or passenger responses. And so we designed a follow-up experiment where we added 2 additional modalities. So now we use a third technology. This is called Excite-seq. It's the 5' 10x technology. We're also now able to get 5' RNA, but also TCR sequences. So for each cell, we know its clonality and what TCR it's expressing. And we added one additional modality where we stain these cells with a dextramer reagent that binds only to the TCR of a subset of viral specific cells. So if a cell expresses that dextramer reagent or we detect it, then we know that it really was mounting an antigen-specific response. And then we get our 175 different cell surface proteins as well. So we're adding a lot of modalities here. We're getting RNA, we're getting protein, we're getting TCR, we're getting this dextramer. We're trying to put all this information together and hope that it'll help us to distinguish between antigen-specific and passenger responses. The last thing that I'll say is we enriched very heavily for CD38 positive CD8 T cells. So that way we got, these were very rare cells, so that way we get a lot more of them in our analysis. All right, so I'm showing you the UMAP here of this follow-up experiment, and it looks a little bit strange because actually now we're actually defining cell state based on 3 different modalities simultaneously: RNA, protein, and also TCR. So cells that are very close to each other on the UMAP will not only have very similar RNA and protein levels, but they'll also have the same T cell receptor sequence. So it's helpful for visualization. As a nice negative control, you can see that our CD8 naive cells don't form these little clumps because they don't have clonal populations. But our effector memory cells, we see lots of these little kind of clumps that are clonal populations. So, I don't want to use a visualization too heavily, but it's helpful sometimes to be able to use this to look at different molecular modalities that we've measured in this experiment. So of course, we measured RNA. So now we can look at the expression of that 200 gene signature that I showed you earlier. And we can see that it is, very highly activated, not in every cell, but certainly in a large subset of them. We can see that overlap is actually quite strong with the dextramer reagent, which we know stains for true antigen-specific cells. And so that's a pretty strong validation that the cells that we had previously discovered are, in fact, at least for the most part, mounting antigen-specific responses. Also correlates with CD38 protein, and then lastly also correlates with the clone size or the clonality of these different populations. So all of this together is telling us that the vast majority of the cells that we had previously identified are indeed mounting antigen-specific responses. But we do see one interesting group of cells here, and I'll just switch to a kind of a clustering view now, that we believe are mounting more of a passenger type response. So we have antigen-specific responses, like I showed you earlier, cells that are proliferating and not proliferating. Those cells are very heavily stained with our dextramer reagent, and they also tend to exist in very large or hyper-expanded clones. As a negative control, CD8 naive cells are, of course, not mounting antigen-specific responses. They're naive, and they're not in clonal populations. But we also do identify this group of passenger cells that looks much more like the naive cells. And so the question is, can we identify, what's unique about these antigen-specific ones versus the passenger ones? And actually, I think one of the cool results from the paper is we see a very nice cell surface protein, KLRG1, that uniquely separates antigen-specific from passenger responses. Both cell populations express similar levels of CD38, which is what most immunologists would use in this context. We identify another marker that we think is valuable for separating these 2 cell groups from each other. All right, that was a little bit of immunology, and apologies for that. I'll go back to some sort of broader conceptual points now. So, now that we've identified these cells with high confidence, in principle, we should just be able to go and find them in any COVID vaccinated data set, including from previous papers in the literature, even if those papers had not originally identified this population. And in fact, that's exactly what happens. If we take data from different studies, now again, we can find cells that uniquely and specifically map to this antigen-specific population. Again, when we have complementary protein data, we can verify the accuracy of our mapping because they exhibit upregulation for CD38 and ICOS. And I'll just highlight that this is exactly why we want to build these references, right? And we want people to be able to we don't just want to be able to discover these cells in our data. We want any researcher to be able to find whether these cells are present in your data sets as well. And of course, we've released all this data openly, so you can map your data to ours and see if you can find similar cell populations. And then the last thing that I'll say is we can also start to look at disease-specific data sets. Everything I showed you up until now was in the context of vaccination, but we can look at disease data sets and say, can we find these same cells in a disease context as well? So here we looked at a data set from the Combat Consortium. So this is a very, very large consortium that looks at hundreds of individuals at different stages of COVID infection and response. And has substantial metadata on how each patient is doing when the sample is taken, but also how each patient is doing as they go forward. And so we were very interested to see if we could find these same cell populations in diseased patients, and whether their frequency or whether their characteristics in some way predict patient outcomes. It turns out we can find these cells very clearly in diseased patients as well. The signature of gene expression is very highly conserved between vaccination and infection. I don't think that's too shocking for us to see, but it was nice to validate that. You can see these cells are very infrequent in healthy controls. In fact, their abundance doesn't vary too heavily with disease severity. So you sort of get a relatively similar number of cells, whether you have mild disease or whether you have severe disease or critical disease. But we did see something very interesting. So the percentage of the cells didn't particularly correlate well with the current state of the patient, but it correlated very well with their future state. So patients that have severe or critical COVID when the sample is taken, a subset of them go on to recover, and a subset of them deteriorate further. And if we stratify patients based on their future state into recovery or deterioration, we see a very striking difference in their T cell responses and the percentage of cells that map to this antigen-specific state. And so again, this is just a correlative analysis. I don't want to claim that we have causal data here, but it's very supportive of the idea that patients who are able to mount a strong T cell response to infection have a better chance of recovering from the disease, and highlights the value of being able to track these responses in patients over time. I'll show you one last analysis, which I think is fun to look at because we have TCR data. So these cells really are memory precursors that we're identifying here. So they will differentiate into effector cells, which will then go on to hopefully kill the virus. And so, we can actually use the TCR of these cells as like a lineage barcode, right? To be able to trace what do these cells turn into over time. And so if we look at patients that have mild disease, we can see that the TCRs that are present in this population are shared with a lot of very highly cytotoxic T cells. So what that means is that in mild disease, these cells are doing their job. They are differentiating into highly cytotoxic T cells, and they're combating the virus. In contrast, if we look at patients with severe or critical COVID, the number of the cells is similar, but who they turn into is a little bit different. We see almost a threefold upregulation away from a cytotoxic T cell fate and towards a much more regulatory T-EMRA fate. And that's really interesting. It suggests that these cells have different fates and are doing different things in the context of different stages of disease severity. All right, so I'll wrap up there and just summarize. And rather than going through the immunology again, I just want to highlight sort of broader conceptual point here. I really focused a lot of the story on a particularly rare cell population that's been challenging to identify in single cell sequencing data sets before, these vaccine-induced CD8 T cells. We found that the multimodal data from CiteSeq was essential for being able to identify and characterize these populations for the first time. In doing that, we could identify both RNA and protein biomarkers for this cell population. And we can also identify this sort of rich gene signature that we can search for in new data sets as well. But once we've identified these cells and confirmed who they are, then we're just going to play the same trick over and over and over again. We're going to try to find these cells in new data sets as well using reference mapping. So first, I showed you that we could find these cells in the TAC-seq data. We found them using the bridge integration technique. And every time we add in a new data type, hopefully we learn something new. So in this case, we identified thousands of enhancers that were unique and specific to the cell type, and also transcriptional regulators that are essential for establishing and hopefully maintaining this memory precursor state. I showed you another couple of modalities that we could add in using Excite-seq, using these dextramer and TCR data sets, and that enabled us to confirm that these cells, at least the vast majority of them, were actually mounting antigen-specific responses, but also enabled us to identify additional biomarkers like KLRG1 that can further subdivide this population into antigen-specific and passenger responses. And I showed you how we could identify these cells in disease data sets as well. To be, there's a very clear conservation of the gene expression signature between vaccinated and disease samples. And that enables us to do a correlative analysis to explore how the frequency of the cell population varies as a function of disease state. And we find that the establishment of this population seems to be very highly predictive of recovery from severe disease. So with that, I'll just go ahead and thank the people in my lab who have done this work. Yuhan Hao led most of the work on the computational bridge integration technique. John Blair led the work on the development of the phosphor-seq technology. And Bingji and Ravi led the work on COVID vaccination. Thanks so much again to BioTuring for having me, and happy to take some questions. Thank you.
Thanks so much, Rahul, for the great talk. Several questions, actually. I've been trying to get the first question out. In some of these slides, you show the UMAP and the original space, not only from RNA-seq, but also from TCR. And how would you put TCR into the distance?
Yeah, so that's a great question, and we actually get that a lot because I think this is something that many users are interested in doing, and actually using most analyses will define cell state on the function of RNA and or protein, and then look to see how TCR correlates with that. In this particular analysis, we define cell state using a combination of RNA, TCR, and protein together. So what that requires us to do, we do that with the weighted nearest neighbor clustering strategy developed in my lab, but now adding in a third modality for TCR, and that does require us to have a TCR distance metric between any two cells. So obviously, two cells that have identical TCR distances will have a distance of 0, but also cells that have similar TCRs will have a closer distance than cells that have very distant TCR. So all you need is that TCR distance metric. I'm actually blanking on the name of the technique right now, but I remember that it comes from Phil Bradley's lab at the Hutch, and we do discuss this in the paper as well. So please do check that out, and if you're interested, you can use the same strategy that we do for defining cell populations on 3 modalities.
Thanks so much, Rahul. The second questions actually from some of the audience, members in the audience. The bridges, I'm trying to reformulate in a way that's easier to understand. The bridges, when you use the bridges to align, one type of data set to another type of modality, does the cell type needs to be present in the bridge to be able to connect between the two sides?
Yeah, so the short answer to that question is the bridge data set needs to be representative of the reference inquiry. So if you are missing cell types in the bridge, that can be problematic. We have seen cases where the method works even when there are missing cell types in the bridge, and the reason it works is because there may be other closely related cell types that can serve as a proxy for the translation. But it's kind of like trying to translate between 2 languages when you're missing words in a dictionary. Like there's no guarantee that it's going to work. So we do recommend that if you want to use the bridge integration technique, that's something that you consider. Often a strong way to ensure that that's likely to be true is to generate a small bridge data set on exactly the same sample that you're generating your query on, because then right, or your reference on, because then the same cell types will at least be biologically present in the bridge. And so that is a requirement. I mean, and I don't think there are good ways around that I would trust, at least at the moment. Outside of relying on the idea that RNA and chromatin, for example, are highly correlated, which I think is also a very risky assumption.
Right. Thanks so much. And also another question related to the bridges. So of course, we have, for example, the bridge has RNA and ATAC-seq, or the bridge has RNA and site-seq. Do we need the number of Cy6 site to be large enough to be connected to the mRNA, or any suggested numbers on how to make the bridges being functional. That means can connect from one modality to another. Is that?
So also one thing also in response to the previous question. So what happens if you're missing a cell type in the bridge? I said it's not good for that to happen. So what happens as a result? So in every one of our predictions, we return a confidence score. So how confident are we that we've been able to correctly map the cell to a cell type in the reference? And so one of the experiments we did in the manuscript was we took a cell type in the query and then subtracted it from the bridge. And we did this over and over and over again and said, what happens? So of course we're gonna map it to the wrong thing because we don't necessarily have it in the bridge anymore. But when we do, the confidence score drops dramatically. So if you see a bunch of cells that are mapping but with low confidence in your query data set, that could be a concern that you might be missing something in the bridge. So we hope that that's kind of valuable and useful to users as they kind of interpret the results from the technique. And the reason the confidence score is dropping is because the algorithm recognizes that it can no longer translate effectively. It's not finding a match between, it's not able to represent query cells as a combination of a missing cell type, for example. In terms of how to maximize the utility of bridges, so actually in basically everything that I showed you today, we used publicly available bridge data sets, and that process does work well. But the best way to be as sure as possible that this is going to work is to generate bridge data, as I mentioned, from the same set of samples. Because that way you really know that it does represent the same biological context. And I think in any integrative analysis, when you have missing cell types or different biological contexts, there's by definition, it will create some challenges. So the easiest way to do that is to generate your own multi-omic data.
Right, and that also part of answering the question from High Volume, asking about should we create bridges data set based on general tissue type or biological condition specific tissue type, right?
It is, and so I think that do you need to create different bridges and different references for different ages, for different sexes, for different disease states? I think that's the question we get quite frequently, and of course the answer is that it depends. Certainly there are differences, for example, in gene expression between older and younger individuals. But I also think that it's very true that we can use data sets that have been generated from younger individuals to annotate data sets that have been generated from older individuals, and then to go back in a second step and look for differences, for example, in the proportion of cell types that arise or changes in gene expression within a cell type. So that's not to say that young and older are equivalent, but it is to say that a data set can serve as a reference even potentially in a new biological context. In the same way, we often map disease data onto healthy genome references. And sometimes there are differences there. Sometimes there are fusions. Sometimes there are deletions. But we can use that initial mapping as a scaffold to help us understand why the new biological context is different. And I think the same thing is true in single cell analysis as well.
Right. Thanks so much. And another, probably related questions about bridges as well is that, the bridges is used for multimodality. Can you use for cross-species?
Yeah, so cross-species analysis is, of course, of substantial interest to us and to others. I don't know how you would use it in a bridge context because I don't know how you would measure a cell that had multiple species together at the same time. There are sort of interesting hybrid models that express, for example, both the human and the mouse chromosome simultaneously, and it would be quite fascinating actually to think of that as like a multimodal sort of experiment. So I think we and many others think about the value of cross-species integration, but I haven't thought about that as much from a bridge context before, but it's quite interesting.
Yeah. So, and how about the different technologies, between query and the bridge data sets? Any study that you have done?
Yeah, so in terms of sort of the size of the computational difference to correct, many groups, including ours and others, routinely integrate data across many different technologies. So as an example, as our sort of primary integration vignette, In Seurat, we demonstrate the data set that we used was a benchmarking data set in PBMCs that was constructed across 7 or 8 different technologies, including combinatorial indexing and microfluidics and SmartSeq and 10x and all of that is integrates, I think, very well, both using our methods and others. But of course, there are challenges. I think in particular, single cell and single nucleus data, in some cases that integrates very well, and in some cases it's much more challenging. And that can be because of actually very significant biological differences between cells and nuclei in particular tissues as well. So I think for many of these questions, there's no uniform answer. It is something where data integration is, of course, extremely powerful and helps you pool information across multiple experiments and analyze them in a common framework and context, which is very valuable. But there always is the possibility, especially as the biological differences in contexts the differences increase, that you may match things together that shouldn't be matched. And I think when we write our papers and give talks, we do our best, especially for key biological results, like I showed you today, to provide some sort of independent validation that the mappings that we are generating are accurate. And so in this case, for example, I showed you, numerous examples with cell surface proteins where we use that as a withheld modality to verify the accuracy of our mapping. And we certainly encourage people to do that, not to sort of blindly trust, whether it's our methods or anybody's, the outputs of a computational pipeline. But those predictions can be extremely valuable. But as with all results, I think require someone to look at and verify and make sure that they make both computational and biological sense.
Yeah. And have you observed any some noticeable difference when annotating ataxic data using 2 different methods, the bridge and also the other approach, the first approach that you mentioned about converting ataxic into gene signal score and then using scRNA-seq as reference?
Yeah. So we do show some benchmarking on this in the manuscript. So for fully differentiated tissues, the previous methods are best suited for, and the bridge integration, I think in every case will outperform, but because it's using more information, right? It has access to more data than the sort of conversion approach. But the conversion approach does surprisingly well, for example, in differentiated brain. And that's probably because there is less of a lag between RNA and chromatin in fully differentiated tissues. In differentiating tissues and bone marrow as an example, it's very easy for that sort of conversion approach to get confused about whether something is a lineage primed cell or a lineage committed cell, because those 2 cells will have very similar chromatin profiles, but very different RNA expression profiles. So in general, if you have access to a multi-omic bridge data set, we certainly suggest that approach, and that's what we've been doing. I think when we wrote the original manuscript in 2019, they really weren't a number of multi-omic profiling technologies, and I think that was a valuable approach for a period of time. But I think to really maximize that accuracy of mapping, especially when you're exploring the relationship between 2 modalities, which is of particular interest, it's particularly helpful to have the bridge.
Thanks so much. Another question is from Nikolai Oskokov. Reference-based approach may suffer from reference bias. That means when the query is forced to be similar to reference. Do you suggest any way to handle this potential bias?
So, we certainly, I think as a sort of broader point, that is true of all supervised analysis, right? Any time that you're going to sort of use prior knowledge, I think in a way to inform your analysis of a new data set, then there's a concern that if that prior knowledge is missing something or is incomplete, then that can be a challenge. And so I think one of the things we really encourage people to do is to try multiple ways of analyzing their data. And in many cases, and perhaps in most cases, we're finding now that reference-based analysis will find new things because it has higher power. If your cell states are in fact well represented by the reference, you will have greater power to be able to annotate cell states at higher levels of resolution. So we're very excited about making these tools and these data sets available. And as these references get better and better and better, that concern about reference bias also decreases over time. But I think as a general rule, we always encourage people you can run unsupervised analysis. You can explore what happens if you analyze your data independently of any reference. See if you come to similar conclusions. If you have very different conclusions, that's something you can explore biologically and try to understand. Maybe there really is a cell type or a cell state that's missing in the reference that you have uniquely discovered in your query. And then that's of course, a very exciting thing to highlight and to support. So I don't believe that any computational method is something that you should just run and trust the results of blindly. But our job, I think, as methods developers is to give you the tools to be able to run approaches that can maximize the ability to discover signal in your data, but never to restrict you from the ability of trying to look at your data in multiple different viewpoints.
Thanks so much, Rahul. The last questions from a high school student. What advice would you give to high school and undergrad students pursuing research on computational biology and modern biology?
Yeah, it's a really interesting question. So thank you for asking and thank you for tuning in. I think there may be two different ways to answer it. One way is to say, computational biology now is a field that is much more accessible, I think, than it was when I was starting out. And what I mean by that is it's much easier now, I think, for someone who doesn't have a very deep background in coding to be able to download data, to explore it, to play with it, both online, but also in R and Python and a variety of community tools. So in that sense, it's possible to start jumping in and getting started to explore and learn a lot sooner than it was when I was a high school student or undergraduate student. At the same time, I think some of the best advice that I can give is, real computational biology, if that's an area where you want to specialize, is more than just taking an existing tool and running a set of sort of tutorials and scripts. It does involve, we emphasize this a lot in my group, is really understanding what methods are doing, their intuition, the math, the statistics, the computer science behind them, how they work, and why they work. I mean, that really comes from reading papers and doing research, and there aren't necessarily any shortcuts to that process. So don't be afraid of that either. If you're comfortable reading papers and trying to explore them and understand how they work, that's really the best way to learn and to explore computational science.
All right. Thank you so much for that advice, particularly for a high school student. And given that, I would like to conclude the webinar today. And thank you so much, Rahul, for the excellent talk. And we have received a lot of questions that I will, send to Rahul later on. All right. Thanks so much, everyone.
Thanks to everyone for staying on for the really fun Q&A as well. I enjoyed it very much.
Yeah, thank you. Have a good day. Bye-bye.